What's going on, guys? Eric here, back with another breakdown video. You guys seem to absolutely love these, so I'm gonna keep making them. All right, we got passage number 4 on the Blueprint FL practice exam. Okay, this is a free exam. Just go to Blueprint, make an account, and they'll give you these exams for free. All right, as always, guys, do the passage first, answer the questions first, write down your answers, and then hear me break it down. All right, see where you went wrong, see where you got lost, all this stuff. Okay, I'm gonna show you guys exactly how to break down these passages and answer these questions. All right, so go through this on your own first, pause it whenever you need to. Okay, this is the first question. Don't worry about this, I filled this in by accident. Okay, this may or may not be the answer. This is question 20, 21, 22, 23, and that's it. Okay, cool. So do it on your own, guys, and then play the video. Hear me break it down. Here we go. The quality of DNA-labeled affinity probes is critical in DNA-assisted protein analyses, such as proximity ligation assays and immuno-PCR. Okay. Efficient high-performance methods are therefore required for isolation of pure conjugates from reactions where DNA strands have been coupled to antibodies or recombinant affinity reagents. Wow, look at this wording here. Do not let this throw you off here, okay? I'm going to stop here and just explain some things, guys. This first sentence, and I would say, yeah, both these sentences are meant to throw you off. They're throwing some words that you've never seen before, and they're meant to make you panic a little bit. But stay cool. You guys got this. All right, I am an MCAT tutor, and I have not seen PLA before. Okay, or pure conjugates isolating coupled to recombinant. No, I've never seen all that. Okay, so you guys got this. Keep going. An important step in the synthesis of these molecules is sequentially removing any unconjugated affinity reagents and remaining free oligonucleotides from conjugation reactions as shown in figure 1. Okay, so they're trying to purify something here. They're trying to purify these molecules, these DNA-labeled affinity probes. And the way they do that is by removing the unconjugated affinity reagents and remaining free oligonucleotides from conjugation reactions, okay? And this is what's going on here in this figure. We don't look at this figure, we only look for it when the question asks, okay? Illustration of purification of affinity binder oligonucleotide conjugates. Do I know what the heck an affinity binder oligonucleotide conjugate is? No. But in my head, it's some sort of DNA probe, okay? And we want to have that probe by itself, okay? I'm going to just look at this for a second here, even though I told you not to, okay? Do not look at this, but I'm just going to do it for the sake of this video. So you have this, and then stuff goes on it, and then you have this. All right, something was added and isolated in some type of way. Okay. First, the conjugation is performed in solution by mixing antibodies with an excess of azide modified nucleotides. All right, so they mixed antibodies with these modified oligonucleotides. Oligo just means few or some. Okay, that's what oligo means. The resulting mix containing antibody DNA conjugates and remaining free antibodies and oligonucleotides is incubated, and the products are immobilized on agarose beads. Okay. Okay, they incubated it. All right. When they mixed it, they put it on beads. Okay. After washes to remove unconjugated antibodies, the captured antibody DNA conjugates along with unconjugated oligonucleotides are both released by enzymatic cleavage using the restriction enzyme MLY1 that cleaves at the border of the complementary region. All right. Between the capture and conjugate oligonucleotides to release single-stranded DNA-coupled conjugates. So something happened here, and then here we got the DNA-coupled conjugates. So the cleavage process required both ATP and S-adenosyl L-methionine in order to run to completion. Okay. The eluate was then captured on protein G beads and washes were conducted to remove any free oligonucleotides having no antibodies attached. Okay, they washed out these oligonucleotides to purify these antibodies. Finally, purified conjugates were eluted from the protein G beads by lowering the pH. All right. And then this is the sequence here, enzymatic cleavage by MLYI. These purified high-grade probes extend the capabilities of traditional immunoassays to allow direct detection of proteins and modifications with high specificity and sensitivity. So this approach here is very, very good. It's good. With them, designated protein targets can be readily detected and localized with single molecule resolution and objectively quantified in unmodified cells and tissue. So they're that good that you can have it localized to a single nucleotide. Okay. This passage here, especially the second paragraph, it can be a little complicated, especially when they go into the azide modified nucleotides and all these details here. Okay. All right. There's times in the MCAT where you're going to encounter passages that make a lot of sense for you and are easy. And there's times in the passage in the MCAT where you're going to be getting lost in these details here. Okay. When these details are pretty complicated, you slow down. You slow down a little bit and go word from word here. Okay. In my opinion, this was worded in a complicated way. They could have worded this a little better, a lot better in my opinion. Okay. But they have a lot of, they're trying to purify. Okay. I kind of have the overall picture. They're trying to purify this thing. They do all these different washes. All right, and they tell me here it's really, really good. And then here they kind of have a figure that goes along with this to help us out, apparently. So how do I feel about this? Do I feel amazing? Definitely not. Do I feel good? I feel about this. Okay. But I remember that the MCAT will throw these passages at you. They'll throw these passages at you that don't make sense sometimes. All right. And they're not supposed to make sense sometimes. They know that it's kind of hard to read these sometimes. They know that the AMC, but they want to see how you do on it. Okay. So let's try to get as most as possible right here. In vitro, free oligonucleotides such as mRNA transcripts have a short half-life in the cytosol. What mechanism is used to stabilize these molecules? Okay. mRNA or pre-mRNA, okay, the poly-A tail is added to the pre-mRNA so that it can survive in the cytosol. All right, that poly-A tail is there, and the cytosol, all these enzymes within, they chew. Okay, they like to chew the end. They like to chew that poly-A tail. Okay, so that poly-A tail is what does that job. It keeps it alive. All right, as well as a 5' cap. So polyadenylation, phosphorylation, no. Okay. Given the locations of the MLYI cleavage site and recognition sequence, MLYI is not okay, they gave you some restriction enzyme types, and then all right, this is very, very, very low yield, guys. All right, don't be upset if you don't get this right. Okay, this is very low yield. I've done this like for 2 days in my undergrad. We went over this really quick. Okay, this is not even in the Kaplan books or anything. I don't think they expect you to know different types of restriction enzymes. So if you want to write it down, this is like some 132 information here. Like if you want a 132, you can memorize this stuff. All right, so I'll go over it with you guys anyways. Type 1, those cleave nucleotides far from the recognition site. So here you have the cleavage site here and the recognition site here. And these are rather close together. There's only like 1, 2, 3, 4, 5 nucleotides apart. So it's not a type 1. Type 1 would be like all the way over here, far, the cleavage site. But the cleavage site is close. So it's not a type 1. Type 2, 3, and 4, those involve the cleavage site close to the recognition site. So it's not type 2, type 3, or type 4. Type 2 also, I'm pretty sure they cleave it within, like very close or within the recognition site. Type 3 is a short distance away, just like shown here. And then type 4, I remember type 4 had to be modified, the DNA, in some sort of way, okay? Don't worry, guys. This is very, very low yield. Which nucleophile was likely used to prepare the illegal nucleotides used in the purification procedure? All right, so how do they get these? What are these illegal nucleotides? First, the conjugation performs solution by mixing antibodies with an excess of azide modified illegal nucleotides. Azide modified. How do they make an azide modified illegal nucleotide? What was the nucleophile here? Sodium hydroxide? No, I wouldn't say it was sodium hydroxide. Okay. CH3CCl? No, I don't think that's it either. ACN? No, ACN is used to create carbon-to-carbon bonds. So it's not the cyanide. It's definitely not sodium hydroxide. That's a strong base. It's a nucleophile too, but it wouldn't add azide. What's an azide group? What the heck is an azide? It's either this or this. This doesn't look like azide. I believe it involves some nitrogens in there. I remember because I would think amide, azide, and I'd be like, oh, those rhyme a little bit. They both involve nitrogen. So I'm gonna go with D. Again, very low yield, guys. Very low yield, but I would know that. I would definitely know that. I believe the azide is the N3. Some proteins and antibodies are susceptible to pH damage. In order to prevent this damage, what would be the most efficient and effective additional step to perform at the end of the elution process? Well, they told us that they were purified and eluted from lowering the pH. Okay. And if we have a low pH and this low pH is damaging these conjugates, and we don't want to damage the conjugates with low pH. Okay, low pH damages conjugates. So we're going to want to raise that pH to something more neutral. Okay, so pH of 3, that's acidic. We don't want it to be acidic. It's already acidic. Acidic is damaging it. 7 of NaCl, maybe. It's still pretty in the low side. Tris HCl 8.5, that's pretty alkaline. That would definitely neutralize this. 13.85, that's way too alkaline. Remember, the pH scale is on a logarithmic scale, guys. So that's freaking huge. 7 or 8.5? I would say 8.5. Okay. 7, it would neutralize it, but it will neutralize it as good as 8.5. All right, we're low pH. We want to bring it up to like high. We want to bring it to like a 7 around there, maybe 7. So this would do the trick better than this one. So I'm going to go with C. Which step of the procedure corresponds to the release of DNA-coupled conjugates? B, C, E, D. Okay, so they have this here. All right, A. What's going on at A? A, they're being mixed together. B, they're binding-ish. C, they're bound. They're bound. D, they're released from whatever this is. This is the beads, I'm pretty sure. D, they're released. And then E, they're bound again. Then F, they're released again. Okay, B is too early. It wouldn't be B. That's when they're getting stuck together. C, again, C is where they're binding to the beads, I believe. It's too early. E, they're going to be bound to these other beads. I think that's the protein G beads here. E is where they're bound to the protein G beads. All right, we go from free to being bound. That's not releasing anything. So I would eliminate E. And D, they go from binding to whatever this is to being by themselves. So they're being released in D. So D is the correct answer here. That's process elimination for you guys. And that's it. Okay. There's better passages in this. Don't worry about this passage, guys. There's definitely way better, but let's see if we got all of them right. Okay. Let's see. 23. got right. That was D. Okay, there's an explanation. 22, we got correct. It was C. Okay, I knew you needed a higher pH there. 7 is, it's good, but it's not strong enough. D, okay, that's the azide. What's an azide? Azide is N3 minus. All right, 20 is A. Restriction enzymes. Okay, if you guys want the explanation, here it is. This is very low yield. And then 19 is A, polyadenylation. So that's the one here. So the A tail gets here, and the cytoplasm chews that A tail up. All right, that's why we need that poly A tail for stability. And yeah, that's pretty much it, guys. So as always, click on the link if you want to schedule an interview session to work one-on-one. Hopefully there's a better passage. Didn't like this one, but it's all right. Let me know what you guys got right. Let me know if it was difficult. It was good practice nonetheless. So I'll see you guys in the next video. Peace.